Hello, everyone, and thank you for joining us on this webinar that is part of SRC's webinar and workshop series. My name is Serge Dandache. I'm the General Manager of Spectra Research Corporation. Our speaker today is Dr. Vicky Broadley. She's the Chief Operating Officer at Nikalyte. She holds a PhD in physics from Oxford University. Vicky has 20 years of experience in the high technology sector in both research and manufacturing environments within global and startup organizations. She has a proven track record for developing new technology for the surface science market, whilst working at Mantis Deposition Ltd, and for consumer electronics markets during her time at Sharp Electronics. More recently, she has worked in the medical devices industry, helping a startup company bring its first products to market. As quality lead for Oxide Limited, Vicky secured CE certification of these products, managed clinical validation and post-market evaluation programs, in addition to setting up a world-class customer care team. In her spare time, because she has spare time, Vicky is a Club Scout leader and an occasional triathlete. The title of her talk will be dealing about nanoparticle coatings, and without further ado, I'll let her go through the presentation.
Thank you very much, Serge, for that kind introduction. Hello, everybody. Thank you for joining us today. I'm just going to share my screen and start the presentation. So hopefully you can see my first slide. So as Serge as mentioned, today I'll be talking about nanoparticle coatings using Nikalyte's one-step process, and specifically I'll be focusing on applications in catalysis and life sciences. Okay, so here's an outline of my talk. I'll start off by just giving a broad introduction to nanomaterials and different methods of synthesis. And this will lead in very nicely to explaining the process that Nikalyte uses in all our technology, which is called gas phase synthesis. So I'll talk about the characteristics of the nanoparticles that technique produces, and also why those are different from other techniques mentioned on the previous couple of slides. I'll then move on to talk about our NL50 benchtop system, which is a one-step process for generating nanomaterials. I'll explain some of the features and how researchers can use the very simple tool to generate a wide range of materials for different applications. I'll then go on to present two specific case studies. The first is using nanoparticle surface-enhanced Raman sensors to detect biospecies. And the second case study will highlight an application in catalysis using high-entropy alloy nanoparticle catalysts for green hydrogen. Okay, so what are nanomaterials? I think everybody here knows nanomaterials are materials in the range of size of 1 to 100 nanometers. So here's a little plot here just to put that into perspective. We're down here in the scale, slightly bigger than a water molecule, but much smaller than a virus. So these are 3D materials. So unlike thin film coatings and two-dimensional structures, which certainly I'm very familiar with, and many of you may have worked with in the past as well, the nanomaterials are three-dimensional. And what makes them really interesting, why do people look at this? Why there's so many people looking at nanomaterials? One of the key reasons is their surface area to volume ratio. So when you get down to the nanoscale, what you're looking at is a massive increase in the ratio of the surface area to volume in several orders of magnitude. And what this means is that the surface becomes really, really important at this scale. So what we see from that is that the knock-on effect for applications is higher reactivity for applications such as catalysis and sensors, which is obviously very important in those areas. We also, not only are we seeing scales of improvement in reactivity, we're seeing novel new properties appearing for materials that doesn't happen in the bulk. And here I've put a nice little picture of some samples that we've produced in our NL50, showing the plasmonic effects on very common materials such as copper, silver, and gold at the nanoscale. And you can see the colors, which is a plasmonic effect due to the size of the nanoparticles, that they appear completely different at the nanoscale to what they appear at the bulk. And it's these properties that we can harness in our sensors, and I'll talk a bit about that later on. Finally, another important characteristic of nanomaterials is that we can use a lot less material to get equivalent performance to 2D and 3D materials. And this is particularly important for areas such as catalysis. And then in my example that I'll go on to talk about in a few moments' time, I'll explain why reducing cost and loading is so important. Okay, so just a little bit of general background into different methods for generating nanomaterials. So there's quite a few different areas of processes available, and they all have their pluses and minuses. They all have their advantages. I'm milling of materials. It's quite an easy technique to scale up, so it's very good for bulk generation of nanomaterials. But it's quite difficult to generate to achieve control. Sorry, not generate, to achieve control with milling. So although it's good for scale up, it's not a great technique for controlling size and properties of nanomaterials. Lithography, on the other hand, is a very precise way of generating nanostructures. And we can do that on two-dimensional substrates using photolithography and other e-beam lithographies to create really, really small features. The main problem is this is extremely expensive and very difficult to scale up economically, although it does still have its place for some applications. Liquid phase fabrication. So chemical synthesis is probably the most common method for forming nanoparticles and does work very well, can be used to scale up. But we end up with a lot of chemical waste. It can be a very difficult process to control. And it's not always possible to generate the kind of materials that you may wish to generate. Pyrolysis is another form of method that can be used to generate nanomaterials. Droplets are formed at the nozzle, but this also suffers from solvent contamination, and there are various chemical and mechanical challenges. But today I'd like to focus on a process called inert gas condensation or gas phase synthesis, whereby the nanoparticles are generated in the gas phase from very pure materials. So let me move on to my next slide. So this is the technique that Nikalyte uses in all of our equipment. And it's a really beautiful technique for many reasons. One of being that we start with a solid source of material, a sputter target here, which is 99.99 or better purity, extremely pure. No chemicals are used, no surfactants, there's no chemical contamination in the nanoparticles. It's purely made from this metal. So let me explain a little bit about how it works, and then I can talk a bit about the features and the controls. So the technique is based on a process called magnetron sputtering. In this image here, you'll see this is the magnetron head here. So we have a target of solid source material, usually a metal or a metal alloy. And we apply a voltage to that target and then put some gas in front of that target. In this case, it's argon gas. And that argon gas is ionized, and those ions basically hit the target and knock off atoms of the material. So you can see these little atoms here floating around inside the chamber. Now, in magnetron sputtering, those atoms will effectively disperse and hit the substrate, and that will form your two-dimensional layer of material. In gas phase synthesis of nanoparticles, we have an additional process whereby those atoms of material are held in an additional chamber, which we call an aggregation zone. And it's in this aggregation zone that the nanoparticle properties are controlled. So we do this by changing the properties and the parameters in the system. So there's various different parameters we can control. We can control the gas flow. So the amount of gas, the amount of argon that we let into that chamber. We can control the power, so the voltage applied to this magnetron. We can then control the gas type. So argon is generally used for generating the vapor, but we can also add other gases. So we can use helium, we can use oxygen or nitrogen if we want to form more complex materials like oxides or nitrides. We can change the distance that the material travels through this chamber. So this distance here from, oh, sorry, I went on a slide. This distance here between the magnetron and the exit from this aggregation zone is effectively equivalent to the time that those atoms of material spend in this chamber. So by changing that distance, we can change the residence time of the atoms and the growth time of those nanoparticles. We can also change the geometry, which is basically the shape of this chamber here. And all of these parameters are controlled by the user. So they're things that can be changed either as the process is running or by mechanical changes before and after the process is run. And they will all impact the properties, the size, the density, the structure of the nanoparticles that are formed. So once these nanoparticles have been through this aggregation zone, they exit, sorry, my keyboard's very sensitive. They exit this chamber through a nozzle, this hole here, into what we call the sample chamber, and they land on the substrate, which is sitting just down here. So at the substrate, the nanoparticles are formed into whatever structure you've made them be inside their aggregation zone, and they land very softly with very low energy of just a few EV onto your surface. So one of the most important features of this technique is that because they land with so little energy, it's possible to coat very, very delicate substrates such as graphene or membranes for catalysis, or plastics, or basically anything that can go in a vacuum chamber can be coated. And that's really important, and that's a feature that's not possible with standard magnetron sputtering. So often this technique is seen as advantageous for that reason alone. So using this process of gas phase synthesis, it's possible to generate nanoparticles with different sizes, range from a few atoms up to around 20 to 25 nanometers, and to control the different material composition and properties. Okay, so let me show you some nanoparticles that have been generated using this technique. So in this example, I've picked some very typical materials that we work with. The platinum group precious metals are commonly used because they form nanoparticles very readily and they're very easy to sputter, but they also have lots and lots of applications in life sciences and in catalysis. So you can see nanoparticles here for gold, platinum, silver, copper, and nickel. And what you'll notice from these lovely TEM images is that we've got really nice crystalline nanoparticles. We can see individual atoms. They're ultra pure, there's no contamination. I've mentioned that no chemicals are required. There's no surfactants. There's no agents required to keep the nanoparticles dispersed. So in these images, you can see we've got non-clumping. Clumping is a real problem in chemical synthesis, whereby the nanoparticles will form in solution, but they just want to stick together. So surfactants are added to that process to keep them from clumping. That's not needed with gas phase synthesis. The nanoparticles are naturally dispersed and they land on the surface and they'll stay where they land. The other feature, which is really key, and I want to mention at this point, is that we have really reproducible results. Because we have such good control over the size, the shape, the structure, the loading, it's possible to reproduce these coatings each time with the instrument. As long as you enter the same conditions at the start, you'll end up with the same coatings, which is really important and is not necessarily the case for other methods. Certainly from talking to researchers working in chemical synthesis of nanomaterials, I know it can be quite challenging to recreate reactions and generate nanoparticles with the same properties. Okay, and finally, just a few comments about the process as a whole. So I've mentioned it's repeatable and reliable. It's easy to produce structures. But one of the other things which is becoming more and more important now is the environmental impact of the process. I've already said that there's no chemicals required in this, so there's no chemical waste. It's also very easy to recycle material. So there will be some waste material. The waste material is basically the remains of a target, a spent sputter target, which is not possible to generate nanoparticles from. And that can be taken back to a supplier and recycled. So the process is really very environmentally friendly. There's no chemicals and it's very easy to recycle waste. So I think that's really important when we're all trying to reduce the environmental impact of the work that we're doing. I mentioned that the nanoparticles are ultra pure and there's no surfactants. We actually have evidence for how clean our materials are, and these high resolution TEM images of platinum nanoparticles demonstrate that very nicely. So these nanoparticles have been imaged with 3 different detectors, a bright field detector here on the left, a high angle diffraction detector in the middle, and a backscattered electron detector on the right. And what they show is the same nanoparticles, first in bright field, which effectively shows us the crystal structure of the bulk. So you can see again, those lovely atomic planes in the platinum nanoparticle. In the high angle diffraction image, you see much the same image, but this image, the contrast is very bright, which we associate with very dense and high weight materials such as platinum. But the image that's really interesting, and I want to draw your attention to, is this backscattered electron image, the last one. What you're seeing here, the backscattered electrons are only giving information about the surface of the nanoparticle. And what we're seeing in this image is that the crystalline structure is present on the surface of the nanoparticle as well as in the bulk of that nanoparticle. And what that proves to us is that that surface really is clean. There's no sulfur or other contamination on the surface. So it really backs up our claim that this is a very pure and chemical-free technique. Okay, so I've talked a bit about the technique. I'd like to now move on to the NL50, our little benchtop system. So the NL50 benchtop nanoparticle system uses the process which I've just explained, and I put a little schematic here just to demonstrate how we fit that into this little neat benchtop instrument. So this has been flipped through by 90 degrees, so you can use your imagination. But effectively, exactly the same process is taking place. We have a sputter target of material at the top here. A plasma is generated with argon gas in front of that, knocking atoms of, in this case, gold off the target. None of those atoms coalesce and agglomerate to form nanoparticles as they travel through this aggregation or nanoparticle zone. They exit through the aperture and then into the bottom chamber, which is our sample chamber, and land on the surface. So you can see that in this image, we've got the top chamber here, which is where the nanoparticles are formed, and the bottom chamber here, where we deposit onto a surface. And the sample, you can see just here at the bottom, inside the NL50 sample chamber. So let me talk a little bit about the features of the nanoparticle system. So the nanoparticle system has been designed to be very easy to use. Don't require users to have any vacuum experience or even any nanoparticle experience, actually. There's preset recipes so that you can run the system. All the pumping and venting cycles have been automated. So that makes it extremely easy to run a deposition process. It pumps down very quickly because there's quite a small vacuum volume there. So in 30 minutes cycle time for doing a nano coating, depending obviously on how much material you're depositing. You can load a wide range of materials. I gave some examples previously of gold, silver, and platinum, other materials as well, titanium, copper, moly. The list is pretty much endless. Anything you can sputter, effectively, anything that's conductor you can sputter, you can form nanoparticles from. We have real time control over loading, which I'll go on to in a moment, and control over size. So let me just click on to the next one. So I want to focus on that control over density first of all. So I've zoomed in here to show you what's the inside of the chamber looks like. So you can see in here, we've got the sample sit here. The example shows TEM stubs, but really you can have whatever substrate you wish. And because it just slots onto the holder, you can even design your own holders for whatever substrate it is that you're interested in. But the part that I want to draw attention to here is this quartz microbalance, which is sitting right next to the substrate. So the quartz microbalance is a little crystal monitor which vibrates and that measures the amount of material landing on it. So during the actual deposition process, we are measuring the amount of material landing on the surface in real time. And what that means is excellent control over the density and loading of material on the surface. So it's possible to generate coatings from monodisperse individual nanoparticles, such as this on the left-hand side, right up to 3D nanoporous layers, which are, microgram densities or even higher. And not only that, we can reproduce this time and time again because we're measuring live as it's depositing. If there's any fluctuations in the deposition rate, we will see that as the process is being run, and we can adjust for it. And you can control the loading. You can actually put in a particular loading that you want to achieve into the program, and the machine will generate that for you. So that's really important that control is present. The next feature that I'd like to highlight of the NL50 is the size control. So I've blown up the touch screen here just to see it a little bit clearer. So it's very simple. There's not many parameters that the user needs to enter, but the parameters that you can enter as a user are the mass, so the density of material on the surface, but also the current to the magnetron power and the argon flow. And the reason this is important is it's those two parameters, the current and the argon flow there, that control the size of the nanoparticles. So let me just move on to the next slide to show you some data from the NL50. So on the left-hand side here, I've got a bit of a schematic here showing the relationship between the particle size and the power or the current on the magnetron and the gas flow to the magnetron. So you can see there's a relationship with as you increase the power, generally the size increases. For the argon flow, there's kind of a peak where the nanoparticle size is largest and then it will start to decrease again. I won't go into too much detail about why that's the case, but rather focus on some data here, which was measured for nickel nanoparticles. And here I've plotted the peak size. So we've got the nanoparticle size between 9 and 1 or 2 nanometers. And I've shown how that changes with gas flow. So we've got for 3 different currents, 3 different powers on the magnetron, we've got trends showing the size of those nanoparticles changing with the gas flow. So these parameters can all be entered, let's say, by the user. And so you can use that to generate nanoparticles of different sizes. In general, we can produce nanoparticles with the NL50 in the range of sizes of 2 to 25 nanometers. I put in brackets that depends on material, because you can see here that for these nickel nanoparticles, we're not getting up to those sizes. It really depends on the material. Some materials are very easy to form nanoparticles, and it's very easy to form larger nanoparticles. Other materials, such as platinum, is a very good example. Platinum likes to form small nanoparticles, so tends to be closer to 2 to 10 nanometers. But again, we have a lot of experience in this area, and we can advise, and there will be inbuilt recipes in the NL50 for certain materials, so you don't need to work all this out for yourselves, and we can help you with any questions that you might have on this. Before I leave the topic of size control, I just wanted to show some TEM images of gold nanoparticles, which have been generated in the NL50. So you can see 3 different sizes here of nanoparticles produced just by changing the conditions. So from 4, 6, and 23 nanometers. And this histogram here underneath the 23 nanometer gold nanoparticles has been taken from the TEM image. So it's been taken after it's deposited. And you can see there's quite a narrow distribution there of just a few nanometers either side of the peak. So just showing the control and the uniformity of size with the NL50. Okay, great. So I hope I've given you a bit of a flavour of what the NL50 can do and what the gas phase synthesis technique can achieve. I'd like to build on that now and talk a bit about a particular example, which is surface enhanced Raman sensors or biosensors. So I don't know if you come from a Raman background, but just a brief review of what that surface enhanced Raman is. So surface enhanced Raman is a technique that uses the plasmonic properties of nanostructured metals, gold, silver, copper as well, to boost the sensitivity of Raman spectroscopy. So Raman spectroscopy is standard Raman is a very nice technique for looking at Raman signal and fingerprints of molecules. Unfortunately, standard Raman is a very weak technique. You only get 1 in every, I think, million photon that comes off the scattered from the molecule is a Raman, is Raman active. But there is a way to enhance that using the plasmonic effects of gold and silver nanostructures. So this is a very well-established technique. Many SRC sensors are available on the market, and most of these are fabricated using colloidal nanoparticles, which have their place and can work quite well. I've shown an image here of a typical colloidal substrate, and what you can see is this problem I mentioned earlier about agglomeration and nanoparticles sticking together in solution. And what happens is when they're either evaporated or dropped onto a surface, they tend to stick together around the node. So you get this kind of very non-uniform coating when a SRC substrate is manufactured using colloidal nanoparticles. And the problem with this is it can lead to poor repeatability because you're getting very different densities of nanoparticles on the surface. The other issue that we've come across with colloidal and chemical substrates is that they can have quite poor specificity. This is due to the fact that there's often quite a high noise background. So although you're seeing those Raman peaks, the kind of weaker peaks are hidden inside the noise. So it's quite difficult to distinguish similar molecules where those sort of weaker peaks are important in identifying what molecule you're looking at. However, with our technique, with physical deposition, we can overcome some of those problems because we have a line of sight deposition, we have very good control over loading, we end up with very uniform coating on our SRC substrates. And I'd like to show some data proving how that can help the performance of SRC substrates for biosensors. So Nikalyte substrates, we use our own gas phase synthesis technology to deposit gold and silver nanoparticles onto glass filter paper. So this image here is a gold coating, gold nanoparticles on, and you can see the fibers of glass. We also have a silver product as well, looks very similar. The nanoparticles have a size around 10 to 20 nanometers, and you can see that beautiful uniform coverage. There's no clumping there at all. There's coverage over all the fibers. And you can see the ones lower down in the structure as well that are coated. And these substrates, we sell these in our online shop, so they're available to buy. But we've been doing a lot of work with partners to test these substrates to different analytes, and that's what I'll like to talk about today. So here's some data for caffeine. Caffeine is a molecule we use quite a lot in-house for testing, partly because it's quite a difficult molecule. It's not massively Raman active. So if we can see a signal from caffeine, then that shows that we have some good material. So what we've seen here and with our testing is we have really good enhancement of the Raman signal compared to in vial. So the black line is just looking at a solution of 1 part per million caffeine. The red line is using our SRC substrate. So we're getting several orders of enhancement for these SRC substrates compared to standard Raman. The other thing which is really important, and I mentioned this earlier, just so I could bring this up again, is the noise level. So what you can see is these peaks go down to 0 in between the Raman peaks. And that's really important because if you want to distinguish Raman spectra for different molecules, you really need to get rid of the noise. And in fact, Raman, part of the power of Raman is really in the database. Without a very good database of clean Raman signals to compare your measured spectra to, it's very difficult to make any sense of Raman signals. So having a clean Raman measurement is very, very important. And the reason we feel that we have a very low background on our Raman substrates is because of those ultra-pure contaminant-free nanoparticles. We have no other chemicals on the surface. It's just gold. It's gold on glass, and that is it. And that's all you see in your Raman spectra. So when you add your analytes, those Raman peaks that you're seeing are just from your analyte and not from any other contamination. And that's really, really important when it comes to performance. Okay, so how have we been using our Raman sensors to detect biomarkers? So I've listed here a whole list of projects that we've been working on with partners for detecting biomarkers. So we've been looking at, our partners and us together have been looking at a wide range of applications here, from cancer to heart disease biomarkers, looking at hormones in saliva, sodium lactate in blood. But the project that I'm going to talk about today is a project that we undertook with partners in America, actually, on detecting the SARS-CoV spike protein. So I will move on to that right now. So this project was actually led by Dr. Wan Kunjia at the Beckman Laser Institute at UCI. So thanks to him for the data. And the project was designed to develop a flexible sensor for airborne viruses. And that sensor could be put into a mask. And then using a laser, you could scan a person's mask to see whether they had a virus. So obviously you can imagine this work started, pretty much shortly after the pandemic. So there was a lot of demand for screening of COVID. But this also applies very much to other airborne viruses such as influenza and any other viruses that might be around. So for the particular experiments that Dr. Gia did, he looked actually at two types of substrate. He looked at the Nikalyte substrate, which was number 2, but he also compared some cellulose substrates, which were also printed with colloidal gold nanoparticles, just to have two different references for the experiment. And in that experiment, the analyte that he was looking at was a spike glycoprotein of SARS-CoV. This was a Delta variant, and he looked at 0.25 milligrams per milliliter in 3 different dilutions. He also did some measurements on a purified, deactivated full SARS-CoV virus. So let me move on to some data. So this graph shows the measurement data for the 2 different sets of experiments. So on the left-hand side, we have the spike protein, the spike glycoprotein with Nikalyte SERS substrates. And you can see that he's looked at 1 to 20 and 1 to 200 protein dilution. And in both cases, we're seeing really clear peaks associated with that spike protein. I haven't put the data here, but actually the cellulose substrate also was able to detect, but only down to 1 to 10 dilution. And the reason for that was that the peaks basically became lost in the noise, which is another example of where colloidal chemical SERS substrates are less sensitive than the gas phase substrates of Nikalyte. So that was a very encouraging result. Dr. Gia then went on to look at the full virus, the full SARS-CoV virus. And in this case, the results weren't quite so promising. We were able to see the full virus, but the signal was much lower than for the standard Raman. And so the problem we think here is that the spike protein is much smaller than the full virus. And so we need to make some adaptations to the SERS substrates to detect the virus as a whole. And that could involve potentially using a tag, like or a functionalized nanoparticle. In this work, the gold nanoparticles were bare, so there was no receptor or binder to bind the molecule to the nanoparticle. And the work is ongoing now, and Dr. Gia's got some new substrates from us is looking at ways to functionalize the nanoparticles to improve the detection of the virus. But this is very encouraging results. The first time we've done any work in with detection of viruses, and proteins. So we're very excited to see this step in the process. Okay. So for my final case study, I'll take a little bit of a change in tact here and start talking about catalysis. So catalysis is a very important area at the moment, and green hydrogen, obviously, I think everybody's heard about the sort of drive to generate technology for green hydrogen and make us less dependent on fossil fuels. So nanoparticles play a really important part in catalysis, and the reason being is that they're ideally suited for use as catalysts for many reactors, including green hydrogen. I've already mentioned that their size makes them ideal. Their surface area to volume ratio is extremely important. And that they also can be used to reduce loading of expensive and very scarce materials. So we all know that there's a problem with the amount of materials for catalysis, and iridium and ruthenium, platinum materials. So looking for alternatives is very important. Also, they're non-agglomerated nanoparticles. That's important because it means that each nanoparticle is active and isn't lost in a bulk agglomerate. And that ultra-pure surface that I mentioned at the beginning, the fact that there are no other chemicals on the surface means that that surface is available for reactions and could be potentially more reactive than an equivalent nanoparticle generated by another technique. The gas phase synthesis also allows us to produce nanoparticles for catalysis from a very wide range of materials. I've already mentioned the noble metals, but actually more earth abundant materials are becoming more important for catalysis. So copper is used a lot in reactions, including for lots of reactions, and nickel as well is another element which is used quite readily for reaction, nickel oxide and nickel iron oxide. The other advantage of our technique is that we can generate complex structures. such as high entropy alloys. And you can guess from the title of this case study that I'm going to talk about that next. So the NL50 can generate elements and alloy nanoparticles from a single target, but I want to introduce another Nikalyte product, gives you another step in terms of degrees of freedom and opportunity to generate more complex alloys. So I've included here two examples of alloy materials that are generated using, in one case, a single alloy target, so nickel iron, cobalt, moly, chrome alloy nanoparticles here generated using an alloy target. So it's possible to generate those with just one target. And in this case, we show that this X-ray image shows that we have each element present in those nanoparticles. So we have actually managed to reproduce the structure or the alloy composition of the target in the nanoparticles. But this isn't always what is wanted. Researchers want to look at different alloy fractions. They want to look at different compositions. How does that affect the catalytic properties? So we have a product called our three-headed source, our DX3 here, which allows the user to load 3 different targets at one time. And by individual control of these targets, generate alloys with different elemental composition. In this case, I've shown an example of nanoparticles generated with 2 different targets, iron and nickel, to form iron-nickel oxide nanoparticles. And that's just one example of the types of materials that can be generated. These sources here, this image here at the bottom here, they are instruments that can fit onto an existing PVD chamber. So the user would need a vacuum chamber of their own. So they are a slightly different option to our NL50, but use exactly the same process. So all the characteristics that I mentioned at the start about purity and control of the size, those are also the case for these sources. So the same advantages over chemical synthesis and the same ease of control is present with these particular products. So to move on to the actual case study, the case study I'm going to talk about is a project which Nikalyte have actually been working on with a group at Oxford University. So Professor Rob Weatherup's group in the materials department bought a nanoparticle source from us a couple of years ago. In fact, this is their chamber here with our nanoparticle source sitting on the side. And the project was designed to look at novel catalysts as a replacement for expensive iridium and ruthenium catalysts for water splitting. And the particular materials that were identified as a cheap and earth-abundant alternative are high-entropy alloys. So high-entropy alloys are near equimolar alloys. So there's no dominant element in that alloy. They all have equivalent ratios. And these constitute from a wide choice of materials, including nickel, iron, cobalt, molybdenum, aluminium, chromium. And these are all very cheap and earth abundant, so extremely attractive compared to the iridium and ruthenium, the platinum group elements. So the particular reaction that Rob's group were looking at is the oxygen evolution reaction. So we're comparing the performance of these alloys to iridium in the water splitting reaction to generate green hydrogen. So the materials generated by Rob's group were, they looked at nickel, iron, cobalt, moly, chromium alloys, and these were generated with different sizes. So we've got TEM images of 2 different size nanoparticles, and you can see they've lined up in these nice columnar structures because they are magnetic materials to some extent. And the team looked at different compositions of alloys. So they looked at mixing aluminium or chromium in with the high entropy alloys, and then performed some electrochemical testing of these materials with different alloy compositions and different nanoparticle sizes. So let's move on to the data. So here we see some CVs for. I hope you can read the text here. So we've got 3 different types of material here. We've got the nickel, iron, moly, cobalt, chrome at the top. We've got nickel, iron, moly, aluminium at the center, and at the bottom we've just got bare nickel. So what was very interesting is that nickel is known to be an active catalyst, but what we wanted to see was how do these high entropy alloys compare to nickel, and are we seeing other elements in that alloy being active, being catalytically active, or is it just the nickel that's doing the catalytic reaction? And what we can see from these CV sweeps is that we are seeing oxygen evolution reaction. We're seeing peaks, we're seeing oxidation reduction peaks, which are not in the same place as just the bare nickel. So we are seeing contributions from other parts of the structure. So that's encouraging. We then also looked at the Tafel slope of these particular reactions to compare the performance of, again, the same three different materials, the chromium alloy, the aluminium alloy, and then the nickel. And in this case, we did see slightly higher Tafel slopes for the high entropy alloys compared to the nickel. But what should be noted is there are differences in size here between the nanoparticles. So it's not clear whether the size of the nanoparticles, which in the case of nickel are 8 nanometers, and the case of the alloys were more like 4 or 5 nanometers, whether that is a dominant feature in the differences in the performance. But in any case, what we were pleased to see here is that the Tafel slope here, this millivolts per decibel, is actually not so different to nickel. And we're quite encouraged by that as a first result. And certainly we're going to go back and look at optimizing these materials further, and also optimizing the experiments to take account for things like the difference in size of the nanoparticles. We also looked at the polarization curves for the two different alloys. So in this case, we've just got the nickel iron moly cobalt chrome and the nickel iron moly cobalt aluminium. And we've compared the effect of size for those two different alloys. And what we see, interestingly, is that the smaller nanoparticles in both cases, so for the chrome alloy, we see that 6 nanometer nanoparticles perform better. In the aluminium case, we see it's the 3 nanometer nanoparticle that are better. And it's not clear whether that's down to basically a effect of the surface area, because in both cases we have the same density of material, the same loading. Not really sure why that's the case, but it's very interesting to see that effect. We also see that the over potentials in both cases are quite promising, and of the order of what we were hoping to see for a OER catalyst. These reactions were done with a fixed electrode setup, and there are some improvements planned. So Leanne took the data, and she's now in the process of setting up a rotating disc electrode measurement, which is more reliable and may be able to give us a bit more insight into the differences between the materials. And we're also looking at varying the composition of the alloys further to understand what it is that gives us the best performance for these materials. But as a first step at looking at high entropy alloys generated by this technique, we're very encouraged. And we'll be continuing this work and supporting Oxford University to generate further results. So just to finish off, thank you very much for your attention through all my slides. I've introduced a technique called gas phase synthesis as a one-step process for generating ultra-pure nanoparticles. I then showed you about the NL50, our little benchtop system, which is very easy to use and can generate these ultra-pure nanoparticle coatings in a very short time. And then I finally finished off by presenting 2 different case studies using this technology, a biosensor case study using our SRC substrates, and also some electrocatalyst results from Oxford University for water splitting for the generation of hydrogen. I hope you enjoyed the talk, and please, if you have any questions, I'd be very happy to answer those. So thank you very much. Okay.
If you don't mind to stop sharing your screen, please. And
yeah, I've just lost there we go. There we go. Lost the button. I hid behind.
So thank you very much for this interesting talk, Vicky. If anybody's interested in asking questions, please feel free to unmute yourself or write it up if you're too shy in the chat section, please. I can start maybe to encourage people. So you described a life science application with SIRS and a catalysis application for green hydrogen. Any other applications that we can think of using this technology?
Yes, lots of applications. I was very tempted to add some more in, but I thought I'd better keep it simple. So yes, so in from a life sciences point of view, we've done some work with antimicrobial coatings, and antiviral coatings, so with copper and silver nanoparticles. So certainly copper is known as being antimicrobial in the bulk, and we looked at coating copper nanoparticles onto wound dressings and surgical instruments to try and make these particular products antimicrobial. And that project was very successful. We definitely saw a high kill rate. I haven't got the data in front of me, so I can't remember the exact numbers. Some of the challenges with that is making sure that the coatings are adherent, because obviously if they're going to be used in contact with the body or with the skin, we need to make sure that those nanoparticles are not rubbed off. So the work involved accelerating nanoparticles to make sure that they were really sticking to the surface, and that was very successful. We also did some work with coating breathing tubes with silver nanoparticles, and in that case, we were able to embed the nanoparticles into the tubes, so that the breathing tubes again were antimicrobial, but there was no danger of nanoparticles being kind of drawn into the tube and into the airways. Admittedly, these tests were done with medical professionals in a research setting and have never been in any kind of clinical trials, but it just showed the possibilities with this technology, so which is extremely exciting.
Thank you, Vicky. Anybody
No problem.
has thought of a question to ask?
Actually, I'd have. Can you hear me?
Yes, we can.
Okay,
Yes.
so first of all, thank you very much for this very interesting talk. I'd have a question regarding the formation of the high entropy alloys you mentioned.
Yes.
I assume because regarding the amount of ingredients, you don't use separate targets for each ingredient, so you have one target.
It can be done in. Sorry, go on. Finish your question.
Okay, so if I assume you're using one target to produce these alloys, have you any mechanism dealing with the different sputter rates of the respective metals? Because you have different sputter rates for the metal, so you don't change the desired stoichiometry.
No, you're absolutely right about that. And if you're using one target, then that's always going to be a problem. One of the things you have to consider with nanoparticle formation is not only have you got the different sputter rates to deal with, you've also got the difference in what we call the nucleation temperature, which I didn't cover in this talk, but which is really basically the ease with which those materials form nanoparticles. So you're balancing both processes when you've got an alloy to try and form a nanoparticle. So not only have you got to balance the amount of material that you sputter, you've got to balance how you form them into structure. So with a single target, you're absolutely right, it's more difficult to do. So we have done hydrogen alloys with a single target and also with our multi-target source. When you have the 3 individual targets, then you can control the sputter yield at each target by changing the power on that magnetron. So in that case, you can get around the differences in sputter yield, and that's exactly what we do when we're forming alloys from 3 elemental targets. For a high entropy alloy target, you're always going to have at least one alloy because we've only have the opportunity to load 3 materials. So in that case, we'd use an alloy of 2 elements in one source, and then the other 2 would be individual elements. So that's one way of getting around it with our triple-headed source. So you're absolutely right. You are trying to fight against the natural tendency of that material to have either a low or a high sputter yield. So that can be an issue, but we get around that by playing with the conditions inside the aggregation zone. So by changing the thermodynamics in that region, by changing the gas flow and the power, and that will change the temperature effectively and the agglomeration. So it is a little bit of a trial and error process. We obviously there's lots of theoretical papers on how to form alloys But really, in practice, you have to do it experimentally to work out how to form particular alloys, particular structures. So that's it. Yeah, very good question. Thank you.
Thank you, Vicky.
Thank you very much.
Okay, I can see there's a couple of questions in the chat. Would you like me to answer those, Serge?
Yes.
Yes. So the first question, should I read it out? So it says, thank you for the excellent presentation. What's the difference or advantage of SRS generated by NL50 over lithography? So that's a really good, it's a really good question. So yes, you're right. SRS substrates, there are many generated by lithography. The main advantage from a commercial point of view is price. They're extremely expensive, and the applications for SRS demand cheap substrates. We're talking to customers working in healthcare, we're talking to customers working in narcotics, and everybody says the same thing. These tests need to be £1 or $1 each. And that's very difficult to do with lithography. With our technology, it's extremely easy to scale up. So we can generate high sensitivity sensors at a much lower cost. That's the main advantage. Lithography does give you really good uniformity, so it's difficult for us to beat that, but we can become competitive and we can become comparable. But as I say, the barriers to usage and adoption of the technology seems to be reliability and cost, and that's where we really can win out on the cost element. So I hope that answers your question. So the person who asked that question, and then
Mr. Choi.
Okay. And then it was just about sharing the PPT, which I'm sure we can do
Sure.
after the event.
In fact, if there are no further questions, I'll thank you, Vicky, and the people who have attended. I just want to say that if you're interested in getting the PowerPoint, it will be more PDF than the PowerPoint or the recording. Just reach out to me. I've on the slide that I'm sharing on the screen, you have my email address. Feel free to reach out directly to me, and we'll be happy to share the recording with you. It will also be placed on our website and our YouTube channel in a few days after some editing. And we look forward to the next webinar. And thank you again, Vicky, for this very nice talk.
No problem. Thank you very much, Serge, for the opportunity.
Bye-bye, everyone.
Thank you.